In this video, we will model DNA replication. DNA replication occurs in the nucleus. We start off with our normal double strand of DNA. First, the DNA unzips. Where the bases meet, there is a weak chemical connection, and the bases can split up relatively easily. The sugar phosphate units stay attached to the bases, but the two strands of the bases separate. A base attached to a sugar phosphate unit is called a nucleotide. There are many freely floating nucleotides in the nucleus. So when the DNA becomes unzipped, the freely floating nucleotides will attach to whatever base they can attach to. In this case, we have a T nucleotide that matches up with an A base. We have another T nucleotide that matches up with an A base. A T nucleotide is matched up to an A base. An A nucleotide is matched up to the T base. A C nucleotide matches up with the G base. And a G nucleotide matches up with the C base. We now have a completed strand of DNA. The right-hand side of the DNA will have freely floating nucleotides attached to it as well. In this case, an A nucleotide matches up with the T base. Another A nucleotide matches up with the second T base. A third A nucleotide will match up with the third T base. The next base is an A, so a T nucleotide will match up with it. A G nucleotide will match up with the C base. Finally, a C nucleotide will match up with the G base. We now have two identical strands of DNA. The DNA has been replicated.